Hello, next is specialized reproductive mechanisms in insects. The majority of insects are oviparous. They lay eggs outside and thereafter entire development takes place. Most of the insects are adapted for laying the only fertilized eggs. Development into embryo from cleavage to larvae often occurs but outside the body of a mother. Such type of phenomenon is called a oviparity. Parity. Okay. There are moreover some exceptions and they are adapted to the different reproductive processes. Various types of reproductive processes are described below, like viviparity. Viviparity is another type of development in which the fertilized egg is retained in the genitalia of mother. Development of embryo occurs on the nourishment supplied by the mother. Viviparity is a characteristic phenomenon in the parthenogenetic Aphidoidea, Strepsiptera, and Puparian diptera. It is distributed widely in large number of insects of various orders. Large number of species of the family Epididae. Then the egg development occurs inside the body of a mother, particularly in the ovariole, in the dipterian, aphid, and chrysomelid species. Then ovoviviparity. In some insects, dipteran flies, musca, larvipara, some other insects, the fertilized eggs are not oviposited immediately and are stored in the median oviduct. The early embryonic and post-embryonic development occurs inside the median oviduct, and finally the eggs containing internally and fully developed larvae are positioned into the outside of the body of the mother. Such kind of advanced oviparity is called ovoviviparity. The development of the larvae in ovoviviparity take place solely on the yolk deposited in the eggs, and no additional nutrition is supplied by a mother. It is in fact the yolk dependent viviparity. The median oviduct in this ovoviviparous insects is greatly dilated and often referred to as the uterus. Other examples of ovoviviparity are recorded from the orders Ephemeroptera, Blattioidea, Homoptera, Thysanoptera, Lepidoptera, Coleoptera, and Diptera. In viviparous cockroaches, Blattella and Diploptera, the eggs are encapsulated in the ootheca. The ootheca is stored in the median broad sac till the first instar nymphs are developed fully. Then the ootheca is held firmly in the female gonopore up to the time of hatching, and then it is dropped outside. In cockroaches, no specific trophic organ is developed for supply of nutrients to the developing embryo from a mother. Then pseudoplacental viviparity. So you need to try to learn about the ovoviviparity in the insect Tachinid Panzeria rudis. Gland, ovary, oviduct, larvae, vagina, spermatica, and uterus. And placenta, follicular epithelium, embryo, amnion, and serosa. These are the pseudoplacental viviparity process. Then pseudoplacental viviparity, this mode of reproduction is noticed in Aphidoidea. The egg is devoid of yolk and chorion. The embryo develops in an enlarged region of the vagina of a mother. In non-pathogenetic condition, the egg is fertilized in the ovariole and the embryo is nourished through placenta-like structures. The pseudoplacentae are formed from the maternal and embryonic tissues. Oral feeding does not occur. In the earwig, the eggs are lacking chorion, vitelline membrane, and yolk. The follicular epithelium of terminal oocyte forms the anterior and posterior pseudoplacentae. Then trophocytes are closely associated to the anterior maternal placenta. Initially, the pseudoplacental cytoplasmic process extends across the amnion and serosa and transport nutrition to the developing embryo, and thereafter serosa and amnion enlarge anteriorly and form the fetal pseudoplacenta. In aphids, the ovariole sheath, embryonic serosa, and endosymbiotes form a pseudoplacenta and supply nutrients to the developing embryo. Then adenotrophic viviparity. In the blood-sucking dipteran flies belonging to the families Hippovosidae. In these insects, the tiny corionated eggs are ovulated singly and the embryo develops entirely on the yolk deposited on the egg. The fully matured egg covered with vitelline membrane, the chorion is transported to the large muscular vagina, often called the uterine, where it is fertilized by a sperm. After some period, the degenerate larva having the anus closed completely is hatched in the uterus and nourished by the special maternal glands, milk glands lying on the ventral wall of the uterus. After full development up to third instar larva, it finally undergoes pupation. The milk secretion is composed of amino acids, proteins, and lipids which are basically derived from fat body or hemolymph. Then Haemocoelus viviparity. In Strepsiptera, the oviducts are totally lacking and the ovaries are embedded in the fatty body freely, and on maturation of the ovaries break down so that the matured eggs are released from the ovary in the hemocoel of a mother where the fertilization and development take place. The eggs do not contain chorion but surrounded by a trophic membrane. The nutrients are supplied through this trophic membrane from the maternal tissue. Hemolymph nutrients are also used for the embryonic development. In Strepsiptera, the fully formed larva hatch out through the brood canal and female genitalia port to exterior. Some Laviparous gall midges also show the Haemocoelus viviparity. In Ciccidiomyides, the larvae first feed on the maternal larval body tissue and then escape through the integument. Then pedogenic larvae of this myster adapted to the Haemocoelus viviparity. Polyembryony. Whenever more than one larvae are developed from a single egg, the reproductive process of this kind is called a polyembryony. It is a common mode of reproduction in endoparasitic hymenopterans and strepsitarian parasites. After cleavage stage, blastomeres independently develop into embryos and are collectively enveloped into a thin membrane called a proamnion, which is in fact formed from the meiotic polar bodies. Entire development of an embryo is carried out on the nutrients entering through the proamnion from the body tissues of the host. A single egg produced large number of embryos, 2 in some insects, 8, 20 to 100 in some other insects. Finally, all embryos develop into the larvae. All offsprings developed from one single cell are often one of the same sex. Polar cells, blastomeres, paranuclear mass embryos, polygastula, polyblastula, polygastula. Polar nuclei, blastomeres, proamnion, paranuclear mass embryos, polyblastula, polygastula. Pedogenesis. If the immature insect named for larva is possessing the functional ovary, capable of reproduction through the pedogenesis, the phenomenon is called pedogenesis. Such precociously matured insects are mostly viviparous as well as parthenogenetic. Some examples of pedogenesis can be cited: the larvae of the gall midges. In Measto, the daughter larvae develop into the hemocoel of the parent larva. They feed on the body tissues of the parent and finally break down body wall of the parent and escape outside. In the gall midges, pupae reproduce the larvae. In some other complex heteromorphosis is shown. Egg produces triangular larva. It molts into the apodous larva, which molts into either female producing pedogenesis larva or male producing pedogenetic larva. Sometimes apodous larva molts into pupa and produce male or female adults also. Each reproductive larva produces vivipariously 4 to 20 young larvae which consume parental tissue before escaping into adopt a phytophagous life. They later on develop either into a first type of larva developing into the adult female or into pedogenetic larva like their parents, or into second type of pedogenetic larva producing a single egg through which develop this male inside this body. Another gall midge have normal male and female and 3 types of viviparous pedogenetic larva producing adult female pedogenic larvae and unlike their parent. Then purely male producing pedogenic larva and the third type of pedogenic larva producing either adult males or pedogenic larva of the first type. In Hemiptera, the last instar larva contains sperm in the hemocoel, showing hemocoelic insemination. The sperm fertilize the terminal oocyte of the ovarian in the larva, and from fertilized egg finally the offsprings are reproduced. Parthenogenesis. Whenever development of embryo from unfertilized egg occurs, developmental process is called parthenogenesis. The parthenogenetically developed insect may be of any sex, but generally with XX homogenetic are the female, or XY or X0 heterogenetic are the males. The parthenogenesis may be facultative when it exists with bisexual reproduction, or obligatory when males are absent, or extremely rare or functionless. The parthenogenesis in some cases has combined in life cycle with pedogenesis and viviparity, or may alternate with bisexual generation. One of the following three types of chromosomal mechanisms is often involved in the pedogenesis. Haplodiploidy mechanism. In the honeybee, fertilized eggs produce females, while the unfertilized eggs produce the males, representing the phenomenon of haplodiploidy. Fertilized eggs produce female, unfertilized eggs produce male. So, apomictic or ameiotic mechanism. In blackheads, aphids, curculionids, and offspring developed from the unfertilized eggs represent exclusively the female sex. They represent the sex of their mother, female sex, strictly, as no meiotic reduction division of the chromosomes occur during oogenesis. Then automictic meiotic mechanism. As plasmids, coccids, etc., the meiotic reduction division of chromosome occur during oogenesis. Both the daughter nuclei fuse together, and as a result, female offspring is produced. Then parthenogenesis in insects is classified into three types. Arenotochy, in which only male offsprings are produced from the unfertilized eggs, Hymenoptera, etc. Arenotochy. Then thaleitochy, in which only females are produced, mostly apomictically and automictically, or due to apomixis or automixis. And then amphitochy, wherein offsprings of either sex may be produced. Arenotochy, thaleitochy, which is females. In other words, the parthenogenetic eggs may contain either a haploid or diploid set of chromosomes and may reproduce both sexes. Parthenogenesis can further be classified into following types. Haploid facultative arenotochy. It is a characteristic of all Hymenoptera in which females laying fertilized eggs develop into females, and unfertilized eggs parthenogenetically give birth to males. This type is also noticed in some coccid strips and beetles. Haploid facultative arenotochy. Then facultative thaleotochy. It is represented by some other insects. Fertilized eggs produce both males and females, but unfertilized eggs fuse with polar bodies during meiosis, become diploid, and produce only females. The facultative thaleotochy is also found in some plasmids. Then obligatory thaleotochy. It occurs quite frequently in some Curculionidae. Exclusive parthenogenesis of its. In the absence of males in progeny, the eggs do not undergo meiosis. Apomictic parthenogenesis, or whenever developed through meiosis, cleavage, nuclear fusion, or oocyte polar body fusion, apomictic parthenogenesis take place. These insects achieve polyploidy in the germline. Then cyclical parthenogenesis. In some species of aphids and cynipids, the bisexual generation occurs in alternation with the parthenogenetic generation during summer and winter respectively. Parthenogenesis may be of obligatory amphitochy type. Hermaphroditism. It is noticed in the scale insect, which besides normal males, hermaphrodite females are present, but true females are always absent. The gonads of hermaphrodite females produce both gametes, spermatozoa, and eggs. Eggs may develop parthenogenetically into the male, and if fertilized in the gonad by sperm of the same individual, then reproduce female, but do not copulate with each other. In the pterophorid and testis, present in the genital ducts opening through separately. Non-functional hermaphroditism is rare in insects. It is reported in the stonefly. Natural castration. Natural castration occurs in insects due to atrophy or developmental abnormality in the gonads, so that they become unable to reproduce functional gametes. Two types: the physiological castration and parasitic castration, as well as origin in insects. The physiological castration is found in several casts of the social insect. In termites, pheromones in the nymphs are present. Sense of epidacil glands and corpora elata in adult stage inhibit development of gonads of both sexes, and the sterile workers and soldiers are developed. In social bees like this, hormone acts adversely on the corporalata of those exclusively feeding on honey pollen only and deprived of royal jelly. This larvae develop into other females.